The aim of this video article is to demonstrate how to quantify the frequency of bona fide neural stem cells in a mixed population of neural precursor cells using the neural colony-forming cell assay. This assay can be used for primary or cultured neural precursor cells from adult or embryonic CNS. Harvested single cells are plated at an appropriate density and then incubated for 3 weeks in a 37-degree incubator. The frequency of neural progenitor and bona fide neural stem cells is calculated respectively by counting the number of colonies that are less than 2 millimeters and greater than or equal to 2 millimeters.
Using the Neurosurgery assay to determine the frequency of bona fide neural stem cells in a mixed neural cell population overestimates the number of neural stem cells. Because there is not a one-to-one relationship between neurosurgical formation and neural stem cells. We developed the neural colony-forming cell assay in our lab to overcome this limitation. This assay can easily discriminate bona fide neural stem cells from progenitor cells based on their long-term proliferation potential. Today we show you how to use this semi-solid collagen-based assay to enumerate the frequency of neural stem cells in cells prepared from dissociated neurospheres from E14 mouse brain. Okay, let's get started.
For this assay we need NeuroCult neural stem cell proliferation supplement, basal medium, neural colony-forming cell assay serum-free medium, and collagen. Pipette 1 ml of complete neural stem cell medium into a 15 ml falcon tube. Take as much volume from the cell suspension you have already prepared and pass through